Okay, so now we're going to go through the last few changes and then I'm going to teach you about pedigrees. Our last video was getting too long, so I decided to cut it for us. So gene penetrance versus expressivity. So in the last video, we were talking about phenotypic plasticity, where the phenotypes can be slightly different, right? And they could change a little bit depending on their environmental situations. There are flowers that if you grow them in the right soil, they're purple. And if you grow them in a different kind of soil, they're kind of a pink. Well, that's plasticity. Now we're going to talk about gene penetrance. Just because you have a gene, does that mean you're going to get this condition? So my family has a high history of cancers. And so my father was always so sure he was going to get cancer. Well, we have lung cancer. My grandfather on my dad's side and my dad both got lung cancer. Am I going to get lung cancer? Do I have the genes for that? Well, I have the genes for it, but it doesn't necessarily mean I'm going to get the disease. So penetrance is how likely is the organism going to actually express the inherited phenotype? I have lots of genes in my body that are never going to express themselves. So if we look at this diagram here, if the phenotype is 100% penetrance, that'd be like thumb, right? Like my thumb being a hitchhiker or not, I have to have one or the other. There's no chance I'm not going to have it. That would be 100% penetrance. But the variable penetrance means that there's a percentage chance that you might show it and you might not. The classic example is something called the BRCA1 gene. BRCA1 gene causes familial breast cancer. So if someone in your family, ladies, had breast cancer, your mom, your grandmama, your aunts, it's good to get this checked. You can actually blood test this. And if you have this gene, you have an 80% likelihood that you're going to develop breast cancer at some point in your life. Not 100%, 80%. So this Angelina Jolie famously knows she has BRCA1. And she made the decision, because she had BRCA1, to prophylactically remove her breasts and have augmentation surgery. So she had her breasts removed so that she wouldn't develop breast cancer, and she had them replaced. Now, insurance doesn't cover that, right? If you get cancer, they'll cover it, but they're not going to cover it like, oh, you might get it. Well, you have an 80% chance, and she had the money, so she went ahead and made that decision. The gene penetrance was 80%. Now, how does the gene express itself? How expressive is it? To what degree does the phenotype show up? If you look at this diagram, it's slightly different. So if you have a narrow expressivity, almost everybody's exactly the same, right? See all the green over there. But if you look on the variable expressivity, that's when you have variety, even when you have the same genotype. Both of these situations, everybody has the same genes, but how those show up is different. So expressivity of a genotype can be influenced by allele combinations, right? Like maybe you have some other genes that cause something different, or environmental factors. The environment plays more of a role in our genetics than we're willing to talk about a lot of times. It's very easy to say, check, 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 I have these boxes. It gets a little more complicated than that, okay? So the last thing I want to talk about, and the last skill we're going to work on in class, is pedigrees and how to read pedigrees. So I want you to look at this chart. This is kind of a key. So females are round, males are square. If you have the trait, it's filled in, so it's affected with the trait. If the individual has died, like sometimes these pedigrees are written for many, many generations, they might have a line through them like that. I don't see those on any of the ones that we usually do in class. Twins, right? They come off of the sibling line together. Adopted, we don't know their genes, right? Their genes are adopted in. And then there's a miscarriage, so the baby had passed. If you have a connecting line between two individuals, we say they're married, or at least they've had children together. The line of descent means this is like the next generation. And so we have individuals here, a brother and a sister. They are siblings, but they are not twins. They did not come off at the same time. And they're in generation number 2. So what do pedigrees show us? They can show us the passing of traits through a family lineage. You can use them to determine how the pattern of the inheritance happens, whether it's dominant, recessive, sex-linked, autosomal. So autosomal traits, if you remember from our sex-linked discussion in the last video, autosomal traits affect males and females equally. So when you look at the pedigree, if the males and females are basically equal, it's not sex-linked. If it's an X-linked recessive trait, The males are going to have it a whole lot more often than the females. And we'll go through some to show you how that works. So here's our first pedigree, okay? Here's our first pedigree. And the first thing we need to always determine is this trait a dominant or a recessive gene? Now I want you to look at it, and I always start at the bottom and I work my way up. If every sick kid has a sick parent, then it's probably dominant, right? It's recessive when two healthy parents have a sick child. So in this case, this specific one is dominant, as there's nobody who's born to two healthy parents with the disease. The next thing we want to know is this sex linked? Is it on the X chromosome or Y, or is it somewhere else? And if you look at this one, the males and females are pretty close. It's like 4-3. And so we consider this one autosomal. Okay, so this one's autosomal. Now we can start figuring out what the genotypes are of the individuals. Remember, if you're sick, you have the condition, and this is a dominant disease. So let's look at individual number 1 up here in generation 1, him. He is sick. He has the condition, so he has to have at least one dominant trait because he has it. But let's look at his children. Individual 1, 5, and 8 are sick, but individuals 3 and 6 are healthy. That means he has to have a big A, little a. What about individual number 6 in generation number 2? What about her? What do we know? She's healthy. And in this case, the disease is dominant, so healthy is recessive. So she's going to have 2 small letters, small a, small a. What about individual number 7 in generation 3? Answer that for me in your Edpuzzle. That's right. That one's going to be small, small as well, because he's healthy, right? He's not sick. If individuals number 7 and 8 have another child, okay, so 7 and 8 over here, have another child, what are the chances that their child will have the disease? So I want you to draw yourself a Punnett square real quick, right? Remember, he's going to be small, small. What is she? Is she a carrier? Does she have one bad one and one good one? Or is she double dominant? That's right. She has one of each. She has one of each because she has healthy kids. So she has to have given her a healthy trait. So she has one of each. So when we get to that last bit of chance, what's the percent chance that the next child's going to have the condition? That's correct. 50%. Beautiful. Yay. Okay. This one, I'm not going to give you all the answers. You're going to have to do it in your Edpuzzle. First question we always have to ask, dominant or recessive? It's recessive. Why? Because individuals 1 and 2 are healthy, but they have a sick kid. Individuals 4 and 5 down here are healthy, but they have sick kids. So it has to be recessive. Okay, sex-linked or autosomal? It is sex-linked. Very good. How do we know? Because there's many more boys than girls. Wait, there's not even a single girl. Not even one. Okay? All right, so let's look at the individuals up here. Now, this is where it gets tricky. Okay, this is where it gets tricky. We have to use X's and Y's to denote gender because we are dealing with a sex-linked trait. So females have 2 X's and males have an X and a Y. The female is going to be XX, but does she have healthy X's or sick X's? So I'm going to give her an X big A, X big A. I'm going to give him an X, she's an X big A, X little a. She has to be a carrier because she has a sick child, right? So she has to have the hidden trait because if dad had the trait, he would be sick. He's going to be X big A and just a Y. The Y doesn't carry a gene. What about individuals number 4 and 5? They're exactly the same as the other parents, right? Mom is a carrier, X big A, X little a, and dad is healthy, X big A and Y. So now, if they have a daughter, what are the chances that their daughter will have this condition? Go ahead and draw your box and tell me what percent chance you think she's got. She has no chance. If her dad is not sick, she can't show a recessive disease. her dad would have to give it to her. Does that make sense? Her dad would have to give it to her. How about individuals 1, 3, and 4 up here? Their mom is a carrier. She gave it to her son, which means because dad and mom both have the trait, the next daughter could get the disease. So as you read these pedigrees, I want you to be able to determine who's got what. I really want you to use Punnett squares to make it better. And we'll talk about this more as we practice in class. Thanks, guys.